Oh, shit! Whoa. Alright, let me get you sorted out. Oh, welcome back! Or just welcome if you didn't see the last video, which you should have done. Yes, we'll carry on talking about how you transform plants. Now, in the last video, we talked about how to transform dicotyledonous plants, dicots, using the Agrobacterium tumefaciens. And that really is the main way on how to transform dicots. Another way is using what's known as a floral dip. That's a lovely name, isn't it? What's a floral dip? Well, one of the standard model plants which we use in DNA technology in transforming plants is the plant Arabidopsis thaliana. So if you call as a model organism in bacteria, yeast in fungi, mice in animals, then Arabidopsis is definitely the model organism in plants. And it does something very, very useful. If you take an Arabidopsis plant, turn it upside down and dip the flowers into your Agrobacterium solution, transformation will take place and the gametes produced by those flowers will contain the gene that you want inserted into the chromosome. So that means you can very quickly produce transgenic seeds which you can plant and form a whole transgenic plant. Obviously you'd have to do the normal self-fertilization processes to get a purely homozygous plant, but we talked about in the last video. Now for the rest of this video I'd like to talk about how to transform monocot plants, monocotyledonous plants. We can't use the same methods as we did for dicot plants because monocots are outside the host range of Agrobacterium tumefaciens. So therefore it doesn't infect monocot plants at all, so we have to use a different method. And I've got to be honest, this method is a bit cooler. It's called biolistic transformation. And what we're doing is we're going to literally be shooting DNA into plant cells. Now this method isn't developed specifically for plants, but it's quite a nice case study to do it in. Of course it's particularly important for us to transform monocots because the monocotyledonous plants actually contain some of the most important plants which we may wish to transform. A lot of the grasses, for example, wheat, rice, that sort of thing. A lot of the plants that we use in agriculture, so to be able to genetically modify them is actually a pretty useful thing. So what you do, you get your plant tissue, put it in a sort of vacuum chamber and then fire DNA at it with a gun, which I've drawn on the top of the board here. This is your DNA gun. Now what it consists of here, I don't know if you can see my writing here, but anyway, what you can see here is a firing pin. Once you set that firing pin off, it sets off this blank charge here. And that converts gunpowder into gas, and that gas sets off this thing here, the macro projectile. At the tip of the macro projectile are all these little micro projectiles here, so little dots here, and that is where your DNA is. It's usually gold particles that have been dipped in your DNA solution. So, blank charge is set off, macro projectile goes hurtling down here, and then it reaches the stopper disc. Now, when the macro projectile hits the stopper disc, all that kinetic energy is converted into heat. So the macro projectile melts and sticks to the stopper disc. But momentum carries these micro projectiles still hurtling forwards, because as you can see there's a little gap here. They go through this gap here and onto your target tissue. So these microprojectiles are dispersed and shoot through the stopper disc at your target tissue. Now cells in the centre of this tissue will probably be completely destroyed just due to the momentum that these microprojectiles are hurtling at it. But once around the side, the DNA within those microprojectiles can be incorporated into their cells. So when these microprojectiles puncture the cell, doesn't cause an awful lot of damage. And once the DNA is in the nucleus, it's all very easy really. Enzymes which are naturally produced by the cell for recombination and repair incorporate the DNA into one of the chromosomes. One of the problems with monocots compared to dicots is that their cells don't work so well in tissue culture as dicot ones do. So what you could have is a culture of these monocot cells in liquid culture, and then you could introduce the right amount of hormones which changes the development of these cells and allows them to make somatic embryos. So what are somatic embryos? Well, somatic embryos are what is the embryo that you would find inside the seed of a plant. So you've got your zygote, it carries on dividing, forming the embryo, and that embryo has the ability to produce the roots and the shoots, and eventually a whole new plant, with the correct hormone treatments, obviously. So this culture is called highly embryogenic. That's because the cells within the culture have a high tendency to differentiate into these somatic embryos because we've got the right chemicals in there. So that's the first gun. Now, it wasn't actually commercialised, and the reason why it wasn't commercialised was because it's a little bit inefficient. The Avon and Somerset Constabulary class it as a gun. So you actually need a licence to have one of these things. So what was actually commercialised is this helium gun here, which is a lot more family friendly, I've got to say. What it contains is this gas chamber, this gas acceleration tube, full with helium. And what you do, you increase the pressure of that helium, and that eventually breaks this rupture disc here because of the increased pressure. Now, below this rupture disc we have a macro carrier, and at the tips of this macro carrier we have, you guessed it, micro carriers. So just like the macro and the micro projectile in the first gun, the micro carriers are attached to the macro carriers, and the micro carriers contain your DNA. They're coated in DNA. So gas builds up in the chamber, it breaks the disc here, that sends this macro carrier with the micro carriers attached hurtling down towards this stopping screen here. Now at that point the macro carrier is well stopped, but there's little gaps in the stopping screen here to allow these micro projectiles to pass through, and they pass through onto your tissue, and some will be incorporated into the nucleus of those cells. And remember, this is all done in a vacuum. And that's it. From that you can create your own highly embryogenic culture like we did in the last gun, and from that you can create a whole new transgenic plant. Now obviously all this stuff about transgenic plants and genetically modifying organisms is a bit controversial with some of you out there, maybe. But we're not going to go into that now. You know, it's all happy days from We're Standing right now. What? Oh no! It's all sad days, isn't it? This is the last video! Oh! Yes, thank you very much. I hope you found these sets of videos useful on molecular methodology. They seem to have been received quite well. I've had quite good feedback, so thank you very much. But for the meantime, I'm hoping to go on to some evolution, which is 10 times more interesting. You know, get a glass of champagne out or something, celebrate the end of my videos, do what I care. I know that's what I'm going to do now. So, goodbye everyone! Ooh.